Welcome everybody. It's great to see that you made it after the gala dinner, in body, hopefully in spirit as well. So we've got an exciting session ahead on emerging technologies. My name's Steve Royal. I'm on the board of directors at Company of Biologists. And before we begin the talks, I need to give you some more advertising about the Company of Biologists. So could I have the first slide, please? So we're a not-for-profit organisation, as I'm sure you're aware by now, which means that the money that we get from publishing and so on, we're able to put back into the community. And we do this in a number of ways. So I'm just going to tell you about some of those over these slides. So we support the community through grants so that people can organise scientific meetings. There are fellowships for travel and sustainability, so they're all listed here on this slide. So these are ways that we support our community. The other ways that we support the community are through funding for 3 biological societies, which you're probably familiar with: BSCB, BSDB, and SEB. They're all present here at the meeting. And we also have a joint meeting programme with EMBO that the company supports. So these are all ways that we provide funding for people outside of the company to organise events and activities, training opportunities, and so on. But also the company organises its own events. So one of those is in the form of journal meetings. So we have 3 journal meetings coming up next year. So one for JCS, one for development, and one for DMM. And there's also a JEB symposium on bio-inspired robotics. So if you're interested in any of those events, please take a look at the website. So these are events organised by the company, similar to the event that you're at right now. The other scientific meeting that we run as a company is the scientific workshops. So this is the programme for 2025. These are focused meetings for about 30 or so researchers to come together and discuss a focused topic and hopefully have some kind of output from that, maybe a white paper or some kind of consensus. It's a mix of senior researchers and early career researchers coming together to discuss their work on a very focused topic. It's a residential, idyllic location, normally in the UK, but I'd just like to point out that we now have one workshop which we hold in the Global South. This year it's in South America on microscopy, and we're doing the same going into the future. If you'd like to organise a workshop, so the great thing about these workshops is that the team at Company of Biologists do all the organisation. So as a scientific organiser, you don't actually have to do that much beyond have the idea and invite some people to come along to the workshop. So I'd just like to draw your attention to the deadline for the 2027 workshops, which is in May. So if you've got a really great idea, please, we'd love to hear it. I'm on the workshops committee. It's a very difficult decision because we get a lot of applications and we can only run a few of these events. But please make my job as difficult as possible. Okay, so those activities I mentioned, the grants for the community, the meetings, and the workshops are only possible because of the work that the company does. So you can contribute to this by submitting articles to one of our journals, reviewing for us, reading the work, citing the work, making sure your institution has a subscription to one of the journals. And I would rather that we do that versus we do all that and profit shareholders at Springer Nature, for example. So, I mean, it's up to you, but this is what we can do with the proceeds of scientific publishing. Okay, we have been running this imaging competition, and they're really fantastic images, and people have voted, and it's time for me to announce the winner. So, drum roll, please. It's number 15, this one here, which is by Oj Ojuk. The first runner-up is image number 2, which is Oh, I don't know what it is, but it's beautiful. That's from Alan Carrillo Boltantano. And the second runner-up is image number 8, which is this one here. That's by Julia Pilogio Di Castro. So congratulations to all those fantastic images. Okay. So what's coming up today? Obviously, we've got this plenary session, and then there's some more activities, so please hang around. We have a lunchtime session, which I encourage you to attend on the community sites that the company organises. There's still a chance to have your say and leave comments in the exhibition hall. Then this afternoon we have a panel discussion, and you can read about it in on the app, but the title is Progress in Science Depends on New Technologies, New Discoveries, and New Ideas, probably in that order. So that's a quote from St. Sidney Brenner. So our panel will be discussing this this afternoon, so please hang around for that. And then we'll be closing up the meeting, and it's been a fantastic meeting. I've really enjoyed it so far. Okay, so now on with the plenary session proper. So it's my great pleasure to welcome Jennifer Lippincott-Schwartz to the stage. So Jennifer is based at the Howard Hughes Medical Institute Genelia Research Campus in the US. She has a long history of using imaging technology to really push important questions in cell biology in a topic close to my heart, which is membrane traffic. So development of photoactivatable GFP, FRAP, multispectral imaging, super resolution imaging, and more recently, correlative volumetric EM, which is really possible at Genelia, and I'm sure that's what she's going to be telling us about today. She's won lots of awards. She's a member of the NAS and won the E.B. Wilson Prize from ASCB. You can look up all the others on her Wikipedia page, but I just want to say that the most outstanding accolade is that she's an editor of JCS, so please send her your papers on cell biology and membrane traffic. And if you'd like to take the stage, Jennifer, thanks very much.
Thank you very much, Stephen, for that introduction. Also, I want to thank the company of Biologists for putting on such an incredible meeting. As Stephen said, I've been an editor at journal Cell Science for many, many years, and it's really been a delight to be part of that organization. And I want to sort of recommend to all of you cell biologists in the audience to send your papers to JCS. It's a really terrific journal. So today I'm going to be talking in this session, which is focused on technology development. And if we could put on the slides, great. I'm going to be talking, I'm going to be sharing with you some new technology that is allowing us to look at some fundamental cell biology at both the micron as well as the atomic scales. And that fundamental biology is protein synthesis. So I want to remind you that protein synthesis is part of the central dogma of biology. It's the process whereby RNA, which has been transcribed from DNA, is translated into protein. And the key core machine of that whole mechanism is the ribosome. And so we're going to hear a lot about the ribosome today, but we're also going to hear about the process by which this incredible giant macromolecular machine is functioning. And I'll just say right out from the start that what its core role is to bring together mRNA and tRNA into this machine to synthesize new proteins. So the ribosome is one of the most evolutionarily conserved machines within all cells on Earth. And in fact, molecules of the ribosome are what really are used for creating evolutionary trees throughout the whole life's kingdom. This is just looking at a cryo-EM of an archaea. And what you can see are all these little structures here, which happen to be ribosomes. So you can see ribosomes are just chock full in the intracellular space of archaea, which are one of the core kingdoms of life. But in eukaryotes, which are more complex than archaea, ribosomes are also found throughout the cell. They're found within the cytoplasm, but also you can see they're studded, frequently studded along the surface of these membranes, which are the endoplasmic reticulum. Now, the endoplasmic reticulum encodes, is basically involved in synthesizing all membrane and secretory proteins within the whole cell. And this comprises about 30% of all of the genome is encoding these membrane and secretory proteins. And this is happening on the surface of the ER through this tight association of these ribosomes with that membrane. There's specialized machinery that is recruiting the ribosome to the surface of the ER. That machinery involves signal recognition particle, which binds to a signal sequence that's being synthesized as a nascent peptide from the ribosome. That signal sequence, which is on membrane and secretory proteins, is recognized, and that brings the ribosome to the surface of the ER, where co-translational insertion drives the peptide into the endoplasmic reticulum, where it will either associate with the membrane or be introduced into the lumen. Now, the key question that we were interested in is, where is this happening on the surface of the ER? When we see all these ribosomes here, we don't know whether these ribosomes are actually engaged in translation. And if you look at the ER from a sort of a whole cell perspective, which you're seeing here, where you can see we've got labeling of the ER distributed throughout the entire cell. You can see that the ER has incredibly complex morphology. It extends off the nuclear envelope out to the periphery of the cell, and it is the largest organelle within the cell, but also percolates throughout the entire volume of the cell. Now, this is just showing using, up here is sort of fluorescence microscopy. Down here are snapshots from a whole cell volumetric EM reconstruction of the ER. And what you can see very easily from this layout here is that the ER has incredibly diverse morphologies. Out in the periphery, the ER is mainly comprised of tubules, as well as these junctional regions, which we're going to be focused on a lot today. But as you move towards the interior of the cell, the ER is taking on these more complex shapes, including we've got these twisted sheets, stacked helicoid sheets, which are found more towards the center here. When these tubules and these junctions get really close, they form what we call 3D tubular matrices. And these can be in 3D or 2D. And then we have flat sheets towards at different places along the, this morphological landscape. So we were interested in where, when, and how is protein synthesis occurring on the surface of the ER. And this is a question that incredibly talented postdoc Hee-Jin Choi was particularly interested in the lab, and he decided that he was going to use some new technologies, some old, some very new, to look at where this protein synthesis is occurring. So how is he going to do this? Well, one way is to tag the mRNAs. And so what Hee-Jin decided to do is to take mRNAs that are encoding membrane or secretory proteins, and we're going to call these mRNAs secretome mRNAs for short, in contrast to mRNAs that are translating in the cytoplasm. We're going to be focused on mRNAs that are translating on the surface of the ER. These are the secretome mRNAs. And so what Hee-Jin does is he takes a particular mRNA that's going to encode a particular membrane protein. He'll introduce these MS2 binding sites. These are basically stem loops in that mRNA that will recognize a coat protein that can bind to this stem loop structure. That coat protein is bound to a halo tag that in the presence of a dye, a fluorescent dye, will glow brightly. When that coat protein binds the mRNA lights up. And so we can see individual mRNA molecules at the single molecule level. And that's what you're seeing in this movie right here, where we've introduced an mRNA encoding cell transferase, which is an enzyme that ultimately will localize to the Golgi apparatus. It's a membrane protein. All of these puncta that you're seeing here represent single mRNAs encoding this Golgi enzyme. Now, if we zoom in on some of these movies, one of the things that Heejin noticed pretty quickly was that there really were two populations of these secretome mRNAs, a slow-moving mRNA population and a fast-moving population, which we've color-coded here in this movie here. Now, importantly, I want to emphasize that each one of these mRNAs that are encoding this cell transferase enzyme are basically localized on the surface of the ER, which we've labeled in the background. The ones that are slow-moving basically led us to start thinking, well, maybe the difference between the slow and fast-moving mRNA pools might be connected to whether these mRNAs are in the process of translating. So to test that, Heejin added puromycin to cells. Puromycin stops translation, and it leads to mRNAs basically releasing from the ribosome and moving around in the cell. And that's exactly what you can see here. When we add puromycin, the mRNAs now release from the ribosome. They're now diffusing very, very quickly within the cytoplasm. And we can see a huge effect of that puromycin treatment. Now, the movement of these rapidly moving mRNAs is very similar, is actually on the same order of magnitude as the fast-moving mRNAs that we're seeing in these control cells. So that led us to speculate that this slow-moving population of mRNA that we're seeing in these control cells represents a translating population of the mRNA. We further found that diverse types of membrane and secretory protein mRNAs are exhibiting this type of reduced mobility on the surface of the ER. So here are different classes of proteins, their mRNAs, and their log mean squared displacement for their mRNA movement on the surface of the ER. And this is in contrast to what you can see for actin, as well as this tail-anchored protein, Sec61 beta, which is translated in the cytoplasm and then later associates with the ER. So in order to prove that, in fact, this slow-moving population of mRNAs that we're seeing on the surface of the ER are engaged in translation, what Heejin did was make another sensor for translation. And this sensor uses a SUNTAG methodology where basically you have an mRNA that can be recognized through its stem loop MS2 binding site. That mRNA is encoding a protein, and when that protein starts translating, we've introduced several GCN4 epitopes, repeated epitopes, in that nascent chain sequence, such that when that nascent chain is expressed, it can be bound by what is called a SUNTAG signal, which is basically single-chain antibodies that will bind to that GCN4 epitope when it's translated. So when you see a SUNTAG signal, you know that this protein, that this mRNA that has an MS2 signal is in the process of being translated. So we introduced this construct into our cells. In this case, the construct is encoding an ER resident protein, cytochrome P450. What you can see is if we just see the mRNA encoding for that protein that does not have a SUNTAG signal, that mRNA, if we do single molecule tracking, is moving all over the place. But if we visualize the mRNAs that have the SUNTAG signal, this green signal here, and do the single particle tracking, which you can see right in here, you can see that the molecules are very confined in their movement. They're stuck in a particular place. So that confirmed that when we see these mRNAs on the surface of the ER, when they're showing this kind of confined motion by imaging, what we know is, what that's telling us is that that's the site where translation is occurring on the surface of the ER. So given that, we started looking more carefully at these sites. And this is where the surprise came. What we found was that the majority of the translating pool of mRNAs on the surface of that the ER is at three-way junctions, or sites where tubules, and we're looking at the periphery of the cell because this is where we can actually see ER structure very clearly. But in that peripheral region of the cell, the translation's happening at these junctional regions. And we see this for many classes of mRNAs encoding different types of membrane and luminal proteins. These are just a bunch of, here's CD4, which is a type 1 transmembrane protein. Saltransferase, which I've shown you, is a type 2. We've also looked at GPCRs, which are multi-spanning, as well as luminal proteins, calreticulin. We're looking at the mRNA here encoding each of these different types of proteins, and all of these mRNAs are localized at these three-way junctions when they are translating. And this is just quantifying the distance from ER junctions that these mRNAs are localized when they are in the process of translating. So we can see for all of those classes of proteins that I just mentioned, they're within about 200 nanometers of an ER junction at any particular time when they are translating. When they're not translating, they pull off to other regions of the ER. This is actin. It turns out actin mRNA can frequently associate with the mRNA, with the endoplasmic reticulum, but it is not immobilized there, and it is not translating there. It's just moving along that surface. It potentially, that association with the ER could have some role in being able to bring ribosomes efficiently to that mRNA, but the actin is moving very quickly along that surface of the ER, not engaged in co-translational insertion. We could also, by single molecule tracking of ribosomes, we're looking at a single ribosomal subunit, we could show that there were multiple populations of ribosomes on the surface of the ER, but importantly, there was a population that was very slow moving, had roughly the same distance from an ER junction, as well as the same overall mobility as these mRNAs. So that led us to say, okay, can we see anything at the electron micrographic level that might support what we're seeing using these live cell imaging approaches vis-a-vis translation of these mRNAs. So what you're looking at here is a volumetric focused ion beam scanning, EM, where we have annotated the ER in blue here, and we've annotated the ribosomes, and what we're showing are those ribosomes that are membrane associated with the ER. Now what you can see from this image is that there is an abundance of ribosomes at these ER junctional regions, and if we zoom up on one of these junctional regions, you can see that the ribosomes have this very interesting sort of spiral array. They seem to be sort of lined up in a pattern that's very reminiscent of what has been described for polysomes, basically a single mRNA that has multiple ribosomes associated with it that are simultaneously engaged in translation. So this really supports our observations from the live cell mRNA tracking that it's these three-way junctions or these junctional complexes of the ER where active translation of secretome mRNAs is occurring. So that led us to ask, well, what's making these ER junctions hotspots for translation? And the core question that immediately came into our mind was, could other organelles potentially be facilitating this? And the reason for thinking this is that we know that the ER makes contacts with virtually every single organelle within the cell. One particular organelle we were interested in was the lysosome. This is just a FibSim reconstruction. You can see lysosomes are very often very intimately associated with the ER. Indeed, we can also see lysosomes associated with RNA granules. So this was another sort of connection that made us start thinking, well, lysosomes sort of like RNA, because they actually are used by RNA granules to hitchhike on microtubules to get to different places within the cell. So there is an intimate relationship of these RNA granules that are chock full of mRNAs with these lysosomes. We further can see that these lysosomes are moving, tracking along the surface of the ER. So this is a grazing incidence, total internal reflection, structural elimination microscopic movie, which really enhances your resolution by twofold over conventional confocal imaging, to look at ER, which is in purple, and lysosomes. And you can see these lysosomes are just scooting along the surface of the ER. The lysosomes are moving on microtubules. In fact, they're the fastest moving organelle within the cell as they move along these microtubules. But remarkably, they are scooting along the surface of the ER, and they're often in very close association with these junctions. In fact, when the lysosome pushes up against a junction, it's sort of stable there for a certain period of time. So that led to the question, could lysosomes in some way be helping regulate the mRNA translation that's occurring on these junctional regions of the ER? So how do we test that? Well, Heejin came in with his tools to visualize translation. So he uses the SUNTAG signal. So we have an ER resident protein that's tagged with SUNTAG and mRNA. So we can visualize mRNA and the SUNTAG signal, which is telling us that this mRNA here is in the process of translating, because that's the nascent peptide that's being made and being lit up. And sure enough, where these SUNTAG signals are localized are right next to lysosomes, as shown in this merged image down here. What's more, the SUNTAG intensity of these secretome mRNAs increased dramatically as you drew closer to a lysosome. So this is the distance from a lysosome and the SUNTAG intensity. So what that suggests is that when you have this mRNA in close vicinity to the lysosome, there's something happening that is triggering enhanced translation. Well, what might that be? Well, maybe it's something to do with what the lysosome is doing. We know that the lysosome is a hydrolytic compartment. It's chewing up proteins. So we look to see whether, if we inhibit the activity of lysosomes, in particular by raising its pH with chloroquine or inhibiting its protease activities with a protease cocktail, what effect that would have on the translation efficiency of secretome mRNAs. And strikingly, we could see a drop in the amount of secretome mRNAs that were being translated under these conditions. So what that suggested was sort of the following. I mentioned that lysosomes are hydrolytic. They have hydrolytic enzymes that will convert proteins into amino acids. Lysosomes also have amino acid transporters on their surface, so they release amino acids. Those amino acids, we think, could potentially serve to boost translation at sites where the lysosome is locally positioned near these mRNAs that are, and ribosomes that are in the process of translating. Now, supporting this thinking, if we deplete exogenous pools of amino acids from cells, under these conditions, cells absolutely depend on lysosomes for amino acid supplies within cells, what we find is an enhanced boosting of that SUNTAG signal near lysosomes. So basically, translation of these secretome mRNAs becomes even more dependent on their positioning near lysosomes for cells to be able to deal with this amino acid depletion. So how are the lysosomes and ER junctions being drawn together? If, in fact, this is the potential role for the lysosome to be in the vicinity of these junctions, what's bringing these two compartments together? And we looked at this protein called LunaPark, which it's an ER membrane protein. It's got two transmembrane domains that, it's got a myristate group. It's also got a zinc finger domain. But most importantly, it's been previously shown to localize at ER junctions. People have not understood why it's localized at these ER junctions, but in fact, that's its most prominent localization within cells. So we decided to see whether LunaPark could be playing a role in lysosome positioning. One way we did that is to use proximity ligation assays to probe whether LunaPark is in close enough vicinity to lysosomes to be cross-linked with LAMP1. And we can see enhanced proximity ligation of LAMP1 with LunaPark relative to other molecules localized either on mitochondria or on early endosomes. We could further show, and this is very significant, this data right here, that LunaPark is marking the sites of secretome mRNA translation. So we came in with an antibody to LunaPark. We localize where what subset of junctions are positive for LunaPark, and then we look at our secretome mRNAs that are high, their mRNA and their nascent peptides are being highlighted with this SUNTAG system. And what you can see is that positive junctions with LunaPark have both SUNTAG and mRNA signal associated with them, whereas junctions that don't have LunaPark, they can have mRNA, but they will not have that SUNTAG signal. And this is just quantifying this data, where you can see that the relative SUNTAG intensity, sorry, this doesn't work too much. The relative SUNTAG intensity is a function of distance away from that lysosome, consistent with there being something very special about the lysosome with coordinating all of this. Now, that immediately led us to start asking what effect on this system, if LunaPark looks like it's important for bringing the lysosome to these ER junctions, what happens if we get rid of LunaPark? And so what you're seeing in this experiment right here is what happens to our SUNTAG intensity distribution if we knock down LunaPark. And what we see is we no longer have a boosted translation signal of SUNTAG at sites close to that lysosome. So we're losing this enhanced translation efficiency that we had seen previously when we knocked down LunaPark. And this is specific for LunaPark. If we look at other ER proteins like CLIMP63 that has been proposed to be involved in controlling translation, if we knock it down, it has no effect on the translation of our secretome mRNA markers. So LunaPark seems to be really important for this. And more globally, if we look to see what the effect of LunaPark knockdown is on membrane protein synthesis through biochemical assays, we can see that whereas the LunaPark knockdown doesn't affect to any large extent the global protein synthesis, which includes cytoplasmic protein synthesis, it's having a huge effect on the amount of protein synthesis of the membrane fraction. So we get about a 50% reduction in the amount of proteins that are being synthesized in this membrane fraction with LunaPark knockdown. So what's the mechanism that might be underlying this effect of Luna Park knockdown? Well, one of the things that we found was a very important protein that's involved in controlling translation initiation, which is this EIF2 alpha, is being phosphorylated when we knock down Luna Park. That suggests that there's impaired translation initiation under these conditions. We further found that overriding this translation initiation control, either by adding a drug called ISREB, which reverses this translation initiation control, it basically overrides it, or if we introduce a different 5' UTR sequence that basically ignores translation initiation control, we can basically bypass this translation initiation defect, reverse it, that were in the Luna Park knockdown cells. And this is just a FRAP assay here that's showing basically what's, I mean, I love this assay because it pretty sort of dramatically shows the effect of Luna Park knockdown in the recovery kinetics after photobleaching one of these sun-tagged spots. So basically a sun-tagged spot represents an mRNA with a bunch of ribosomes on it that are translating a single mRNA species. If we photobleach that spot and we watch recovery, we're watching translation initiation and elongation basically happening. That's the recovery process, and in control cells, you can see that recovery within 100 seconds. But when you knock down Luna Park, the recovery never gets complete, and it takes much longer. Importantly, if we add the drug ISREB, which basically reverses the depletion of the integrated stress response, the activation, it inhibits the activation of the integrated stress response and overrides translation initiation control, we bring things back to normal. So overall, what this is telling us is the following model. Basically, the presence of lysosomes at these junctional regions basically boosts translation of membrane and secretory proteins by, we think, providing a ready localized pool of amino acids for charging of tRNA molecules. It's also recently been shown that the lysosomal surface has tRNA synthetases associated with them, including tRNA synthase that it codes leucine. Leucine is one of the major proteins that's found in transmembrane domains because it's nonpolar. Basically, the idea would be that this translation machinery on the surface of the lysosome is being brought close to sites on the surface of the ER via the role of Luna Park that then facilitates translation initiation on the surface of that ER. Now, I want to, in the remaining few minutes of this talk, move to the atomic scale to share with you some very new technology that I think is moving us into the direction of actually seeing dynamics of atomic structures. So I mentioned that we can see ribosomes very nicely in EM, but the problem is that protein synthesis is a dynamic process. Basically, tRNA is continually, charged tRNA is coming into that ribosome. It's docking on various sites along the mRNA. It's then moving, and a nascent peptide is being synthesized. This is a hugely dynamic process. The question is, can we make a movie of this process in atomic detail? Now, we're moving in that direction from cryotomography, which has provided static snapshots that are suggesting structural changes to the ribosome during translation. Basically, we have a rough idea what's happening to the conformational states of this ribosome as you move through the elongation cycle. But the issue is, can we get enough of these structural states to be able to see in detail this elongation process. And so I want to share with you some work that we've been doing recently using a different technology than cryotomography, but it's using cryo-EM, and this is high-resolution template matching. And so basically in this approach, which Peter Rickauer was really one of the key players in developing, he developed this in collaboration with Winfried Denk when he was at Janelia, and Peter then came to my lab, and we moved it into this arena of basically imaging at the atomic scale to make movies. So basically this approach is you take a high resolution noise-free template of a macromolecule, in our case it's going to be a ribosome, and we search for it exhaustively in an image acquired at minimal EM, at minimal electron beam exposure. That's the real issue with cryotomography at this point, is that you're hitting your image with an electron beam multiple times. Every time you hit your specimen with an electron beam, you are destroying structure, and you want that structure. So in this approach with high-resolution template, we have very minimal electron beam damage, where detection is not dependent on the abundance of a target or averaging. Basically, we're making use of this high-resolution signal in a noisy image to try to get structural states. So basically, what we're going to be doing is we're going to take cells, put them down on grids, gold grids. We'll do high-pressure freezing. We'll take a picture. In this case, we're taking a picture at the edge of a cell, because we're going to be just taking one picture. We're not going to do a tomogram. We take one picture, 200 nanometer thickness at the periphery of the cell, so we don't need to do any kind of FibSim shading. And we're going to start looking at that structure. We're going to search that image exhaustively for a molecular target. And the target we're going to use is a ribosome. So we know from classic work, 30 years, the structure of that ribosome has been characterized. So we know we can use it as a template. In this case, this is the large subunit. We can simulate what that template is going to look like in our EM image, and that's what you see to the right. And then what we're going to do is just rotate that image in every single direction to see if we can cross-correlate anything in our image that we have acquired at the periphery of the cell. And this is just, that arrow points to a cross-correlation hit that comes up as beautiful signal to a beautiful signal over the noise. So we know that at that precise spot in that image, in our image, the ribosome, the large subunit of that ribosome, is positioned precisely in the way that we have up there. So we can do this and map out all of the basically hits for not only the large subunit, but we can look at separately the small subunit body and the small subunit head. And you can see that based on the size, the small subunit head, the signal to noise is a little bit less, significantly less than what you get with a large subunit. But we can visualize all of these components of that ribosome. And when we do that, we can see precisely where a full, completed, assembled ribosome is, and where parts of the ribosome are localized within that frozen sample. Now, this is really significant because, as I mentioned, that the ribosome, as it moves through the translation cycle, is basically the different parts, the large subunit and the two small subunits, are rotating and swiveling relative to each other. We can precisely map out those swivel and rotation angles because we've identified every single ribosome in the cell and the position of all of these subunits. And so this is just showing the sort of population of conformations of all of the ribosomes that we've seen in the cell. If you look at the far left, each of those ribosomes, the swivel and rotation angle between the large and small subunits are being plotted in a conformational space that correlates with the elongation cycle state. And to the far right here, you can just see the abundance, the fraction of ribosomes that we're seeing in these different elongation states. And I should just emphasize that the stars just show you examples of states that weren't previously seen using in vitro approaches. Now, one of the things that we can immediately see in this kind of work is when we look at a single polysome, we can actually see correlated states of these ribosomes in a large population of these polysomes. We could show that these correlated states do not represent colliding ribosomes, because the interface of of the two ribosomes that are close to each other does not match with what has been shown with colliding ribosomes. So what we think instead is what this is representing is that the ribosomes along an mRNA, these polysomal arrays are actually in synchronized states, similar to the way that rowers are synchronized in the way they're moving through their various elongation and transformations to drive the boat through water. Now I want to just, I know I'm totally off time, right? I'll just quickly just show that in addition to being able to look at these different states, we can reconstruct molecules that are surrounding that ribosome using this approach. Basically, we just look at electron densities. Once we've found these ribosomes, we look at electron densities in the regions of those ribosomes, and we can then use existing structures to match those electron densities to identify different proteins. Now, I want to just show you that we can take we've got a whole slew of different states of these ribosomes. We've put them into an order, and basically you're going to look at the translation process. We've got 41 different states of these translation, of the elongation cycle that we can line up and basically play as a movie, and that's what we're looking at right here. So the blue is the large subunit, and then the brown is the small subunit, the body and the head. What you're looking at are particular molecules that are associated with this ribosome, and the purpose of this is you're going to watch as the ribosome moves through that elongation cycle, what happens to these molecules. They're coming on, they're coming off. We can see three fundamental types of motion. We can see rigid body motion in this case, this is a 28S ribosomal mRNA. It's basically staying associated with that large subunit as it swivels around. We can also look at proteins that sort of connect the large and the small subunits together. This is the EL19 and EL24 molecules, and these show a bending motion, which had not been seen before. Basically, as the two subunits are moving relative to each other, this linker is bending back and forth. We can also see the tRNA as it moves into the ribosome groove to sort of start building the polypeptide, undergo what we call spring-like motion. So with that, I just want to end by saying that we think this high-resolution template matching potentially provides a roadmap for looking at in situ dynamics. This can easily be done with other molecular complexes, and we were able to show that we can get down to a size limit of about 300 kD, kilodaltons. And we also, we can see that because you can generate these fine-grained 3D time series, you can start looking at mechanical transitions to this mechanical motion. You can combine high-resolution template matching with classic single-particle averaging approaches, which are used in cryo-ET. You can combine these different techniques to get even broader information about these macromolecules. And finally, we're very excited to potentially use this approach in conjunction with AlphaFold to look at a range of molecular targets for detection in cells. So with that, I want to thank everyone in my lab that was critical for this. I mean, Hee-Jin Choi, shown up to the left in the red arrow, and Peter Rickauer, right arrow. Those are the key people in this work, but I also want to thank the rest of my lab, but also key core groups at Janelia that really play fundamental roles in enabling us to really push this technology. With that, thank you so much for your attention.
Thank you, Jennifer. That was absolutely awesome. So the next speaker is Manu Prakash. I think when we were putting this session together about emerging technologies, we were thinking, you just saw absolutely cutting-edge technology, multi-million-dollar, available to very few people, and actually Manu is famous for frugal science, which is putting low-cost technology into the hands of many thousands of people. So two extremes. I think he's going to talk more about his work in ocean science, but I'm looking forward to hearing your talk, Manu. So if you'd like to come to the stage. Thank you very much.
So I brought the tools with me if anybody is interested in playing with them. Maybe at lunchtime, I'll be around. These are two of the objects. I don't know if I'll get the time to talk about them. There's roughly around 3 million kids right now walking around with microscopes in their pocket, exploring and observing our world. And one of the things that I want to spend some time on is really talk about what does it mean when we distribute access to all of the scientific tools that we so much care about. I'm going to start my timer. So one of the challenges that I often have is to stay focused. So this is going to not go into the depths and the details of any individual discoveries, but just give you a flavor of how we do science. There is a whole bunch of destinations that I have planned to take you on, and you get to choose where you might want to go. So first of all, because I know I won't finish my talk, incredible number of people I get the chance and honor to work with. These are alumni of the lab, the current members. They are brave, they go out in the world, fierce, harsh weathers, and pretty difficult working conditions, and still make something out of it. So I'm very proud of the group. If you don't take anything away from the talk, there are two things I do want you to remember. One is, when I think about company of biology and just biologist, I love the fact that they keep the word biologist. It's not ecologist, it's not cell biologist. Very rare for me to be at a meeting that I can go from one session to the other, connecting all length scales. And one of the favorite children's book that I want to share, but it's also my favorite book, is this book called The Giant Golden Book of Biology by the famous illustrator Charlie Harper. The book is out of print, it's very hard to find. But what I love in that image is you notice that the lion and giardia and single cells are all drawn at the same scale. And I often think about the tree of life is every single organism is an idea. And as often, and we heard that in the very first talk with the degradation of our planet, something that we love so much, we're actually losing species probably faster than we can find them. And this false divides that we have created between many of the fields, I want to break that a little bit today, and I'll tell you about just what does the discovery, if you could throw a dart, not to kill any of the animals, but land somewhere on a tree of life and just work on that organism, what does that look like? And I'll tell you one way of doing that. The other thing I want to talk about emerging technologies is technologies are good when you have them. These two pictures, this is Heckle out in the field. What I love is he's wearing his portable belt-tight microscope. He was in Sumatra at this time. Of course, many of you know about his work in cell biology and radiolawrence. Just remarkable tools, but often we think about taking these technologies to the field. Go where the biology is. And I know many of you have been thinking about this in the context of ecology, but I'll tell you, what does it feel like when you take a confocal microscope to high seas? And then the most important technology that I would say that we're all carrying but we've sometimes forgotten is the technology of wonder. And this is why we work with children, and I'll talk quite a lot about this. This is also my second favorite children's book. Okay, so why work in an ecological context to begin with? Why not just stay in the lab and do the work that you want to do? And I think that's totally fine. This painting it's called the Drewer's Rhino. It was made by a Portuguese artist. It was the very first representation of rhinoceros, artistic representation. But this artist had actually never met a rhino. Stories from India propagated along through people talking about it. You know, he got some things right, but rhinos don't look like that. And I feel sometimes in science that can happen when we forget the ecological context of why organisms, and I'll show you examples of cells without the ecological context you cannot understand their any shape or form or ask sets of questions. So I feel we don't want to make this mistake. The last 5 years for us has been 17 expeditions around the world. So here is also a menu. Where would you like to go? I could take you to a swamp. I could take you to the polar Arctic. I could take you in the middle of the ocean, much of the blue ocean. We could also go to the beach. And I'm serious. You guys think that I'm joking. Where would you like to go? Everybody wants to go to the ocean. Okay, let's go to the ocean. Actually, let me take you to the beach and I'm going to take you to the ocean. No, I could also take you to the ocean. I'm tempted. Okay, I will take you slowly to the ocean via the beach. When you arrive, when you're ready to embark and you sat on the sand, you stopped for a minute and you picked up some sand. That happened to me in Santa Barbara almost 10 years ago. And I saw something under a foldscope, which any of one can use. This object, the most beautiful object that I've ever seen in my life. What you're looking at are sea cucumbers. The reason they're polarizing is because they have these incredible stones in their body. And actually, the object that I saw under a foldscope was this little anchor on the very right. This is a foldscope photograph, and I just couldn't believe myself. I had thought humans had invented anchors. It's not true. And it kind of, they're rolling around, doing their thing, and often enough you get stuck and just starstruck with beauty. 4 years passed, we tried every year trying to grow sea cucumbers in the lab. It's damn difficult. They only spawn once a year. We have a spawning season. We only have around a month of live juveniles, but we succeeded in the end, and I'm going to tell you what we discovered about patterning, and then I promise you I take you to the deep ocean. This is what we saw. So the object to the right is essentially the rocks that were inside the body of the sea cucumber, and on the left is the exact same mineral in composition. This is calcite. So nature makes that, but nature also makes that, and it's been an outstanding question for the longest time, how mineral structure is programmed. Where are the developmental cues associated with this? And again, one of the threads is just a lot has been said about the beauty of this system. Peckel, many others drew these structures. In fact, many of the sea cucumbers that we know today that are extinct, we only know them from these sets of structures itself. And in general, biomineralization is everywhere. I wouldn't be standing here. I would be plopped down on the floor, succumb to gravity if I wasn't mineralized. And in some sense, a lot has been said about function. I'm not going to talk too much about function because we know nothing about why sea cucumbers make ossicles. But in other places, eyes have been built. You can sense gravity. You can stand. There is a context of thinking about carbon sequestration in the ocean that's deeply connected with mineralization, and a wonderful poster on how mineralization can actually protect plants itself. I also learned foldscoping one day that we also cough needles. We cough mineralized structures. I don't know why, but if you ever image your cough, you will actually find mineralized structures in them. Mineralization has also been very interesting from a climate perspective recently because people have been able to image forams. These are single cells that were collected in the HMS Challenger and collected very recently. And you can see literally the change of pH in the body of these organs. These are fossils that have been recording the chemistry of the oceans. And one of the challenges, and I'm just going to jump to the specific thing that I want to talk about, is the reason we chose sea cucumbers is we wanted to really understand the cell biology of mineralization. We know somewhat about bone formation. We know quite a lot about magnetic mineralization, but just really how do cells shape these sets of minerals? So on the right are mineral structures from sponges, and on the left is sea cucumbers. And if you notice something very interesting, just evolutionarily itself, the only difference between these two sets of, a big difference has been topology. The fact that sets of organisms figured out how to build topologically complex structures. So whenever I use the word topology, think of your coffee cup. Coffee cup is genus 1 topology because there is one hole. Donut is also a genus 1 object. And I think the thread for us has really been, for the last couple of decades, the idea in mineralization has always been you will find this one specific protein and it's going to completely change and program, or maybe a cocktail of proteins. And that's worked out to a certain extent. Frankly, chirality, for example, can be controlled by amino acids. There are certain sets of ideas, but shape programming has really remained an open question. And these are two ways that ossicles are not made, but I want you to remember that some of the mineral structure and architecture, the most well-studied problem which we do understand is actually in snowflakes, and I'll tell you that this is not how it works. A brave graduate student in my lab decided to join this project, and tireless 4 years of work. We tried all kinds of species. We called all kinds of people, especially restaurants that eat or serve sea cucumbers, were very helpful in teaching us some of the skills. But in the end, we were able to actually culture and keep this species alive in the lab. And what you're seeing here is an organism that's alive and completely transparent. So I'm just going to show you live videos of how these mineral structures are made. They're made very slowly. So first of all, we can label them with calcine. You can see these remarkable structure in a juvenile, roughly 300 or so stone-like structures. Now, what's important is that they're truly single crystals. So we can grow and you can measure the crystalline axis and you find that the crystalline axis is indeed preserved. They're not amorphous structures like for some sets of systems like diatoms. With help of Rudolph, we actually measured quantitatively the orientational dynamics of these structures itself. They are always pointing outwards, one of the axes. So something is aligning the normal axis of the body to the crystalline axis itself, which remains a puzzle. And now with micro-CT, we can essentially map every one of these sets of crystals, do a lot of topology on these sets of structures. I'm just going to focus on one specific structure in the middle. There are several different classes associated, but that's the most abundant structure. And one of the things that's fascinating is there's a lot of geometry, there's a lot of topology associated with this. We can try to understand ideas around errors, because when you write in stone, you don't get to correct errors. So you can see the seed structures when they form an error, it propagates. And one of the threads around this is the idea of finiteness. How do you actually program some sets of crystals encode shape, other sets, as in footpad, grow as the animal grows. So there is a paradigm associated with how these structures are finite. And finally, really the mathematical sets of structures of how do you break symmetry in these sets of systems. So again, I have a rule for any sets of question, just go and watch. And we built a microscope that allows us in a live organism to actually watch a single ossicle grow. And what we see is a cluster of cells, and right in the lumen of that, a tiny little crystalline seed. The seed has certain kind of symmetry, and you're going to now watch that symmetry breaks, and that acts as the seed of these sets of structures. So you can see the same set of structure in the same animal, but you have symmetry errors. There's a trisymmetry, quad symmetry, pentasymmetry, 6 and 7. Majority of them have that quad symmetry. And I'm just going to show you what this process actually looks like. and we ended up discovering this is branching morphogenesis. So this was 1 micron per hour, so it's a pretty painful process, but these are the 2 key steps. You can see branching and you can see fusion. Now the process became much more exciting when we actually look at the cell biology of this system. One of the things we can do is we can study these sets of structures, we can map the entire growth, but who's making these structures? And when you label these live cells, what we find, these clusters of sclerocytes, these cells wrap these rocks like a saran wrap. And in this TEM, you can actually see a 40 nanometer thin cytoplasm shaping the rock itself. So in some sense, one big question to think about, which is a much more conceptual question, when somebody made David, you could actually see it. If you were a sculptor, you can see it. How does the cell know what it's making? And the simple answer to that is that the cell becomes the sculpture. By wrapping this object completely in this conformal way, the shape of what it's making and its own shape is actually one and the same. And then one of the key factors that we discovered is these sets of positional cells essentially secrete calcium in these vesicles, deposit them on the surface. Much of that deposit is done on the tips, for example. So this is somehow these vesicles that you see up on the top are being transported to the tip and always deposited at the tip, and that's where majority of the growth is. Now, what would program those sets of vesicles? So first of all, you can actually watch these vesicles live. This is all done in live animals. And you can see the transport, there's incredible motor proteins involved in this, but kind of the image that I want you to remember is the road network. The cytoskeleton of these cells essentially programs a road network, these are microtubules, on which these vesicles are moved along. They're deposited at these sets of tips, and in some sense to think about, the structure in the past programs what the microtubules would look like in the present. The present microtubules lay out what the road network would look like for the future by laying out the rocks. So this is this very constant feedback between the living architecture, which is the microtubule, and the dead architecture, which is these rock-like ossicle structures that determine the shape. For some actin fans, actin is involved in the fusion, very similar to growth cones in neurons, actually. And one of the big things that we find is that there are actin projections at the tip that search, just like neurons searching themselves. And that search leads to a fusion, leads to a membrane bridge, and that's where the fusion for these sets of structures begin. You can watch these fusion bridges, and over time, these sets of fusion bridges grow. And since I'm out of time, I just want to show you, when we put all of this cell biology together in these mathematical frameworks, we can now simulate these types of architectures. And this has a massive phase space, and I don't have time to go in. But a real surprise came in, and this is also just the power of mathematics sometimes. So this is flux on a graph network. As the graph network changes, flux changes. Flux is determined by the microtubule. And this was one example of a structure that we discovered that first showed up in our simulations. And then we looked through the entire database of sea cucumbers, and we ended up actually finding an ossicle that kind of looked like, that just gives you an idea of thinking about these sets of correlative sets of notions. There's a lot of work that we're doing on the molecular underpinnings of these sets of structures, nanosims, being able to actually do the single cell seq, and also methods to be able to determine errors associated with these sets of systems. But I don't have time for that. One of the things, I will now take you to the ocean, is we went to the Arctic looking for exactly the same sea cucumber that Haeckel had drawn, which happens to make these wheels. And we now have, we don't have them live, but we can actually still see the same sets of microtubule architectures on these wheels itself. This is what that species looks like when it's alive. Okay. So since I already brought you to the Arctic, let me just slowly from the Arctic take you to the open ocean. So let's go to the polar worlds. You heard a lot about the polar worlds in the first talk, so I'm not going to review this. We're in a pretty terrible shape. Look at the movies about how much ice we've lost. The reason we planned this polar expedition was we were interested in species that live in ice. And what do? And one of the factors is we've been working on thinking about what does the nucleation of ice look like? And does life play a role in nucleating new ice? Because as you know, ice is dynamic, it goes back and forth. Could there be a biological parameter that's associated with it? And I don't have time to talk about this, but we have discovered a biological parameter, a single cell that actually nucleates ice. So just what does it feel like to being in the Arctic and working? This is one of the drones. Now this is very thin ice, so it's actually quite scary, primarily because we can only drill thin ice, and we are interested in the interface between the living ocean and the ice. The first surprise, which was known from a prior expedition in 2011, but we re-recorded it, is the highest biomass under the sea ice ever recorded for plankton or life in the ocean. 100 times higher. Now this should come as a shock because under the ice, light doesn't penetrate. But because we're perturbing these worlds, ice is thin enough that enough light is penetrating. There's plenty of nutrients, so we have these massive blooms that have never been seen by satellite. We actually don't know their total capacity for carbon sequestration itself, which is why we originally planned the expedition. But I will tell you some side discoveries. One of the big discoveries that we made is we found many species of diatoms living in these ice structures, and we were very curious, are they just trapped there? What are they doing? And we had built, this is what the photographs look like. There is a ton of life in sea ice. Ching, one of the postdocs in the lab, decided to essentially do this assay where we had built a very, very cold microscope that can go down to minus 20 degrees Celsius. And lo and behold, these diatoms literally just glide around. This is ice. Now these are ice channels that they're moving inside, so we've been able to image these ice structures. And what's very exciting is now this diatom holds the world record for the coldest temperature for actomyosin motility. This is an actomyosin-based motility, minus 15 degrees Celsius, and they're completely fine. And there's a lot that I could tell you about this myosin, which I don't have time for. But one of the key things that we were able to actually do and show is many of these diatoms are actually adapted to these sets of temperatures, and we can compare these sets of data sets amongst many different diatom species to show the key sets of adaptations. There are two key adaptations. One are ice-binding proteins. Many marine diatoms that are not from the ice cannot bind to the ice, so they cannot generate these traction. They generate these tractions by making these beautiful mucus tails, that's the actin cables. And one of the things we've also been able to do is to do traction force microscopy and measure the velocity of this mucus threads being generated. So you can watch that in the raphe. And in some sense, this is what the traction force microscopy on this looks like. One of the key aspects of this from a point of view of adaptation, when you think about this, is, the planet has had a lot of snow cover. Different people would argue whether we had completely snowballed or not. So this should not come as a surprise. But once we lose the kind of ice cover that we're talking about, we also lose a remarkable number of species. And frankly, just like the biodiversity shift is happening, many plankton, tropical plankton, are now moving up, further up, taking over these new landscapes. Now, there was another surprise that happened in the Arctic. I want to give sort of a remark for many of the ecologists in the room. Whenever we go out, we try to look, and this was a brilliant idea of two of the graduate students, Pranav and Ethan. I've been quite concerned with just the conversations that are happening around mining the ocean, and we wanted to just understand what is at the bottom of this ice. So we just took a pretty cheap rover, a couple $1,000s. We actually lost the rover in the middle of the expedition, but we got the data. Otherwise, I would not be able to show you this video I'm about to show you. We plunk the rover, and you're now diving deep right under the ice cover. This is in the Chukchuk Sea. It's close to the Russian border, but it's not in Russian border. And this is what you see. I will talk about the little drab things that you see. This, I feel like this is the, whatever that movie, Star Wars. You're going in, and then boom, on the surface, we find the largest ever collection of brittle stars. kilometers, as far as we go, wherever we put this, just an incredible assemblage of these remarkable organisms. And then when we went back, looked at the data, 60 years ago, people had reported this is a single species of brittle stars, all organized, sitting there in the dark, under ice, for at the least we know 60 years, because that was the last reporting. And one of the fascinating things that happens when you look, you look at these assemblages, you ask yourself, what are they doing? They seem to be holding hands. We've done a lot of percolation networks in this. We can show this is a percolated network. I can't prove to you that they're talking or information propagation is happening, but something quite remarkable we discovered. Took this data, we quantified it to hell. This happens to be the Aphera species. One of the things with this data is that we can essentially build these very large mosaics and statistics. And from that, we realized something quite remarkable, that these benthic communities are organizing themselves in what are called hyper-uniform network. Hyper-uniformity is a beautiful idea in physics between crystalline structures and completely random structures. For example, if any of you study the retinal cones in chicken, there is a beautiful series of papers that have shown how many of the cones in the eye of a chicken are organized in a hyper-uniform structure, because it's the most efficient way to capture photons and light. And ironically, the little drab things that you saw, these brittle star networks are essentially catching, that's their food. And this hyper-uniform network that we can show that this is actually the scale there shows that these are hyper-uniform networks, is primarily the most efficient way that they could be capturing food, because most of the time the only food that they have is through marine snow. Okay, so now once we've, I'm actually quite excited about now reanalyzing some fossil records as well. I don't know whether we will ever find true fossil assemblages because we would like to know how old is this assemblage in some sense. But we also have an experiment in the lab to see could we recreate some of them with another species of brittle stars. That's on the way. Okay, now let's actually go to the open ocean. And now I'm also going to go much, much faster. So how many of you have been to the open sea? Just so that, raise of hands. And no point raising hands because I can't see anything. But I see a few of you have. It's a remarkable place. It kind of takes your breath away. It's kind of scary. But frankly, we know very little about this. And so this is why we go. If you think about the numbers, and just from a context of climate, and I want to, in the last few minutes, I want to connect cell biology and climate change. You see, that's the top curve is us pumping more and more carbon into the ocean. The bottom curve, that's called the ocean sink, is the ocean responding to this increase and soaking in more and more carbon. So every time you have breakfast, you should thank the ocean twice, once for giving you half the oxygen, and the other for taking half the CO2. Between 30 to 40% of all anthropogenic carbon is being soaked in. 90% of all the heat is being soaked in by the ocean. And one of the irony is that we don't know how that process works. So talking about 10 years ago, when we started hearing about CDR, I got quite concerned because I didn't believe we understand how this works. So much of what I will show you is how that works. And probably one of the largest errors we have found in our estimates of how this ocean sequestration works. So first of all, I'm not as brave as these two people. I cannot even imagine just putting myself in a steel ball and just jumping down. But the most famous thing William Beebe did, of course, everybody knows him for bioluminescence in the ocean, showing that life existed. He broke the world record for being in the deepest waters. But on a telephone call, when he was there, while Gloria was sitting on the top of the ship, he said, "It's snowing down here." And everybody thought that, oh, oxygen is too low. They're kind of going cuckoo. It can't be snowing down there. But in fact, he was referring to marine snow. And 60 years passed, nobody cared about it, until a time we actually learned that that's the mechanism for how ocean sequesters carbon. One of the challenges that we have is the ecosystem as an ocean, and the biological pump is one of the most intricate ecosystems. It's an aquatic ecosystem, tens of thousands of, this is not filtered seawater, this is live native density of an ecosystem on a night dive. So, when you ask yourself, how do you understand? If you were to shut down this pump per se, the biological pump for sequestration, our carbon would go from 400 parts per million to 600 parts per million instantaneously. So this is a very important pump, and people have been thinking a lot about how this works. There has never been a quantitative approach, so we asked ourselves to just go measure, because we were concerned that there are very few biological parameters. And I'm just going to show you how this works. The image that I want you to take is a chasm in the ocean, because the ocean is 4 kilometers deep. You want to understand the vertical context and the technology that we built for it. And because I know, so first of all, why we should be concerned, that prior to this work, this was the state of affair of how we essentially balance gravity-driven sedimentation of this living ecosystems. I could draw any line in that curve and fit that to see what the balance would look like. And that's why we have errors. We have to do better. So the technology that we built is primarily an infinite field of view vertical tracking microscopy. So a way to think about it is, if you have an organism or this marine particle sinking 4 kilometers, you want to remain in its frame of reference for infinite distances in the vertical axis. I went to my chair and I wanted a very large building so I could build this microscope. We did actually build a 5-meter tall microscope, by the way. But on one of the trips going to Madagascar, we had this realization: what if the two ends of these tubes were connected? That led to the very first hamster on a wheel, but for single cells. We call this machine gravity machine because it has a giant dinner plate which is made of glass. There are 2 locations, 3 o'clock and 9 o'clock, where you can image. And we reverse whatever an organism does. So if a cell is going down, we spin the wheel up. If the cell is going up, we spin the wheel down. This is a hydrodynamic levitation mechanism, so in your own frame of reference, you can image something forever. I'm just going to show you lots of videos of this tool. This happens to be a coral larva. We currently have the world's largest behavioral data set, roughly around 650 species, collected in the native environment. Temperature, pressure, light, salinity, all of these sets of parameters, to just understand what does life do in the ocean, before we could answer how does it essentially sequester carbon. Much of this data is published, so I'm not going to show it. I want to jump to this carbon sequestration side of the story. One of the big things we discovered is gravitaxis is universal in the tree of life. So almost all the sets of organisms that we see, actually I should show you one beautiful picture. Much of these videos are online, but you're watching a sea star larva, and it seems as if it's stationary, but it's climbing 0.5 meter in this movie. And one of the things that we can then understand is the kind of behaviors that are possible that we can study in this context. So going back to sequestration. So first of all, many of you study cell density. Cell density is a remarkable parameter. We know nothing about cell density in the ocean. On average, cytoplasmic density is 5 to 10% heavier. We took a diatom, this is in Alaskan waters. You think it's sinking because it has glass. Within 100 milliseconds, it's changed its density by 10% to be completely stationary. This is one of the fastest density change ever recorded in a cell. It's not attached to a wall, it has no bounds, and it has a mind of its own, and it decides to go, and it just does this all the time. So life in the ocean regulates where it is. Now, that's piece number 1 of the puzzle. Now, what ends up happening is you also have cells, and especially in the context of cell cycle, where they will choose to do a certain position. Cell cycle is synchronized to the depth in the ocean. This is a bioluminescent dinoflagellate that we discovered that goes 0.5 kilometer deep in the ocean to divide and then rise back up another 0.5 kilometer, 7-day life cycle. If you divide this 1 kilometer travel by the size of the organism, it's the largest migration of any kind and also the largest cell migration. But the clever thing that the object does, and one of the things that we like about talking about reviewers, I'll make a comment about reviewers. We have 100 meters of this kind of climb, and you have a 100 micron cell. And I was really excited because Adam, the postdoc in the lab, he came up with this term of a whale bar. And so instead of scale bar, we have whale bar in these pictures. So please use whale bars. The reviewers were very unhappy with us. One whale bar is 30 meters, so if you need it. So here is the surprise. This cell has no cilia, no appendages, but massively inflates. When I say inflate, 6-fold volume change of a single cell to essentially, so that's, you can see that within 10 minutes. It's the largest volume change I am aware of a single cell. And I know volume homeostasis is a big deal right now. This is not 1 or 2%. This is 6-fold increase. This is massively ballooning. And if you don't believe me now, this is on a traditional microscope, and you see this massive inflation. Since I don't have time, I'll tell you about the cell biology of this inflation. But the point is that it's important to understand that all of these living organisms are essentially controlling where they are, what they want to do. So now, what does the ensemble do? Much of how carbon sequestration in the ocean works is dead cells, live cells joined together to make these living ecosystems that then sink. This sinking is really what saves our planet. And we were, for the first time, able to collect large enough data sets for these sinking debris, and we noticed something quite troubling. And I'm just going to show you what that is. So I'm just going to choose, first of all, on the right is sinking velocity. On the left is many of the marine snow particles collected in the Gulf of Maine. You see there is no correlation between these sets of structures. And this was a correlation. A Stokes' law is used as a correlation in many of our calculations, and we showed that that's completely wrong. Because these objects are living, and for a living object, you really have to think differently. And the reason, very simple reason, it actually turns out, and I'll play this video carefully if you watch, you notice this particle sinking, but behind it you see this massive transparent mass traveling. These are polysaccharides generated by the bacteria that populate this set of a structure, and they generate parachutes. What we believe is that these bacteria don't want to go to the deep waters. They have their food, and they have come up with a way to slow it down. And slow it down to an extent that a 200 meters per day sinking particle could go to 0 meters per day. So absolutely shut down all flux, essentially. And one of the things that happens because of this, if we now take all the data from 5 of our expeditions all together, so first of all, these mucus tails are quite common, we can show that the total mass that's associated with this is an 80 gigaton error in our current calculations. So you can now see the modified Martin's curve. And all of this is biological. We have no clue what these polysaccharides are made out of. We don't as yet know which sets of bacteria. Many bacteria are involved. But the most important thing that I can tell you is without accounting for these biological nonlinearities, it'll be very difficult for any of us to be able to perturb, forget about CDR, just by what we do, there will be incredible nonlinearities. I know I'm out of time. On the technology front, I just want to say 2 things. We're now building an in situ machine that allows us to watch this deep in the water. For the first time, we have actually now been able to also label all the bacteria. So to truly show you how living this is, now this is in the open water. You can watch, this is the native density of bacteria, by the way, in the ocean. And you can watch these organisms zipping by, feeding, colonizing this, and essentially making it a living ecosystem. Let me just say one sentence about doom and gloom and kind of recovery. Doom and gloom is we are literally dismantling all our capacity to do science out in the field. Marine stations are under decline. Many of the capacity of being in the ocean is under decline. The only marine station in the entire bay is actually shutting down right now. And one of the things that's been our approach to this has been democratizing technology. There's a couple of sets of tools, if any of you are interested in engaging. The first one is called Planktoscope. This is an instrument that you can build with your students, your kid. It's right here on the floor. Cost us roughly around $500 to $1,000 to make, and people in their garages build this, and this has now become an incredible time series of aquatic ecosystems. There's roughly around 500 communities around the world. All of the data essentially goes to a common server, and we built this first for sailors, but it's become a massive network of aquatic ecology. And then the second set of a tool that I want to mention and talk about, which some of you know about, is the Foldscope itself. This is responsible for a lot of discoveries that I actually talked about, but it's an instrument that's a microscope, cost us $2 to make. We ship it for $2, and this is the type of data sets that come out of that, single bacteria resolution at that price point. At this point, there are 3 million kids walking around with this tool. These are citizens of the planet that, as much as you and I love nature, but have never had the opportunity to engage. And one of the reasons it's become one of the largest microscopy community at this point, all the data goes online, there's a lot of taxonomy involved in this, but the most important part is that the network is completely distributed. Everywhere from juvenile prisons to public libraries, to nomadic communities, to ocean goers. The network belongs to the people. So there is no centralized operation. Nobody works for anybody. It's almost science at the service of citizens rather than citizen science. Lots of data is collected. It's completely open. Anybody can analyze. There's roughly around 1,500 papers written. Lots of species of tardigrades have been discovered. Some chickens have used microscopes. Fishing communities, elephant diseases. This is the Foldscope Club in Iraq, right after the war ended. We do a lot of work in refugee camps around the world. I want to close with this picture. It's my favorite picture. I talked about many sets of things. We can manufacture technologies. We can even get them to people. We cannot manufacture mentors. And what I'm asking many of you to think about is, if you deeply care about curiosity and wonder being an answer to some of our problems, take the initiative and spend time with kids. Take many of these tools or other tools. What I love about these parents is they're not sharing the microscope with their own child. Be that parent, but be that parent for everybody. When you do science out in the open, when people observe that in a train station, many of you are taking a train to London, set up a microscopy booth right in the train. I'll have some Foldscopes. Play and share. And I think one of the things that we don't realize is the power of how infectious wonder is. And to a certain extent, I know that it's not the entire answer, but I do believe it's one of the answers. And I'm sorry for taking a little bit longer. I'll end here.
Thank you very much, Manu. That was absolutely fantastic. So we're going to take a few questions on the stage. I think if I don't ask any of the questions that you've submitted, there's a lot of you, we might get lynched. So let's do that, and then we'll take the refreshment break. So Jennifer, if you'd like to join us on the stage as well, we'll move on over. Okay, I'm going to take this one first. So this is to Manu. How common in the developing ossicle is asymmetry or error?
So I think because it's a topological object and you cannot correct errors, if the symmetry itself has an error, that remains, and that symmetry error is not that common. But topological errors are quite common. So if even the stones and ossicles that look similar are not the same genus, they're not same topologically, because as this process grows, any fusion mistake would lead to the entire trajectory being changed. So if you were to define error by topology or genus number, that's very common. But some of the other traits are still preserved. You can have a topological error, but the ossicle is still the same size, which is quite intriguing. So that has to do with the flux terms. The total number of sclerocyte cells determine the size, because that's the amount of calcite that they can synthesize. So I think the word error should be thought about in the context of function. We don't know anything about the function, but the form, roughly, we can say that certain aspects of the form are preserved and others are not.
So the next question both of you can answer, which is, what important technology, given that we're at the Biologists 100 anniversary meeting, what important technologies still need to be developed for the field which might emerge in the next 100 years? Jennifer, you'd like to take that easy question?
I'm not. Yeah, it's one of the things that I think that's really interesting is just if you look at the way technology develops, it's in many senses sort of unpredicted. You know, like the super resolution technology really would not have happened unless there was a GFP technology. And a GFP technology would not have happened unless there was some understanding of microorganisms or jellyfish. So there's a sort of a interaction among all of these technologies that you really can't predict. Things happen. I mean, people jump in to a field once they see something, and then depending on who's jumping in, things start emerging. I think one of the things that I certainly think hard about is what's going to happen with AI and the way that it's going to make our lives easier in certain respects, in the sense that we're going to collect lots and lots of data, but it's going to be very challenging to make sense of that data, and you know, that I think is a huge problem that we have to be thinking about. But yeah, I'll just pass it on to Manu, because I know that he's going to have some very interesting perspectives too.
No, I think predicting the future is just a mistake. One technology that I would like to exist, not that I don't know whether it'll happen, is, you know, we all want to study Gaia and this social organism that we live on. What if a social organism was studying the social organism? And what I mean by that is truly studying science as a community. You know, when you talk about ecological and planetary sets of problems, we don't have enough scientists on this planet. And even to an extent, the kind of initiatives that we've been able to create for some of the most urgent problems don't have the kind of cohesiveness that's needed. So I wish a technology to exist where we really disrupt. I'm not saying for everything that works, but for some of the largest scale planetary problems, we need a way to study it in a manner where we are a superorganism and a superbrain. And no matter what has happened so far, I haven't seen that play out.
Okay, we'll do two more, one to each. So have you considered, this is to Jennifer, have you considered visualizing neuronal cells to capture local translation in neurons that are actively firing?
Yeah. It's in progress, basically. Yeah, I mean, we're definitely interested in it, and there are things that you have to sort of do in preparation for something like that. You need to really understand clearly where these organelles are. We recently found some very interesting structure of the ER in neurons, in particular dendrites, that we think is really important for the way calcium signaling is being propagated within that system. And that's going to be really important for interpreting, you know, lysosomes, mitochondria, and how translation might be regulated in that system. But it's something that we're really interested in.
Progress. Okay, thank you. And Manu, do you think the C-star network is a survivor's effect, where stars that are not in the network cannot get enough food and die?
Yeah, I think there's lots of physiological things that we have been contemplating. The one that we know for sure is that this hyper-uniform network is the most efficient way to capture falling rain of this organic material. with the least amount of competition. Now, one of the other things that also happens is that new organisms are born and they die, and we don't as yet have the kind of time lapses that we need to how much of the system reorganizes. So, for example, we don't do perturbation experiments primarily because this ecosystem has probably been there for hundreds of years. But long-term monitoring and effectively, these are very large-scale ecosystems, spanning kilometers. What is happening at the periphery might also be really interesting. We don't know how the density happens at the chain. It's just unfortunate when we were at sea. We have to be very careful about which territory we are on when we park the boat. And so we cannot cross certain territorial waters. But I really am itching to see what the edge looks like. And I think the edge would give us a lot of answers associated with how an ecosystem essentially progresses slowly.
Okay, so thanks everyone for submitting questions. I'm sorry I couldn't get time for all of them. We need to go to the break now, so it just remains for me to thank both speakers for a truly magnificent session today. Thank you.